to receive here at the Institute of Computing Dr. Samir Velankar for a lecture. Dr. Velankar received his PhD at the Indian Institute of Science in 1997 and was a postdoctoral fellow at the Oxford University, UK from the 1997 to 2000. Currently, Dr. Velankar is a team leader at the European Bioinformatics Institute, EBI, at the UK, where he's a member since 2000. He was involved in a number of very relevant, very important developments in bioinformatics. One of the major ones includes the development of the AlphaFold model. And Dr. Samir is here in Brazil this week for the X-Meeting plus BSB conference, which took place in Curitiba, Paraná, starting tomorrow until Friday. And fortunately for us, he's making a technical visit here at Unicamp, and he will present to us a lecture entitled PDBe-Knowledge Base and its role in facilitating structural biology research. His talk is co-hosted by RECOD, the Artificial Intelligence Lab here at IC, and also by the Centro de Biologia Molecular and Engenharia Genética, the CBMEG, we can start.
Yeah. Thank you. Thank you very much. This is my first visit to South America, so it's been great. I think people are really nice, and actually I invited myself, so I wasn't supposed to come here. I just wrote an email and saying I am coming to Brazil. Can I come here as well? And they said okay. They said okay. So I think it's a great pleasure to be here. And what I'm going to present today, I think, is how the structural biology, bioinformatics, and in general life science research is changing, and what kind of things we need to think about in making sure that the structures and the models we have actually impact the translational and basic research going forward. Okay, so right, so I want to start the story in 1971, and there is a really good reason for that. This is the time when the PDB was established with 7 structures, and this is the first open digital database for any biological information. So previous to this, there were databases in chemistry, but this was the first biological database that made data open access. So just the vision of the data becoming open access and it having impact. The people who started PDB, they knew what they were doing. Right? I was just born, so nothing to do with this. And I think from the very beginning, the idea was this is a global effort, and everybody should participate and make sure that all the experimental structures, wherever they are solved all across the globe, should be deposited in the PDB, and they should be openly available. So nowadays people talk about all this FAIR and open access and all that. None of those fancy words, but that's what this is. And that's what actually was followed by many other data resources in making their data accessible. It's now managed by the WWPDB consortium, which has 5 partners, and we now have PDB China as an associate member as well. So this is again making sure that this is global effort and everybody can contribute equally. Okay. So as these structures became available, you could immediately see the impact of those structures. So if you see very early on, this is a seminal paper from Chotia and Lesk in 1986, and with very, very few structures and very, very few sequences, they realized that the sequences change more often, but the structures are a lot more conserved. And obviously the the implication of that is you can use structures to model sequences which are different as long as they are very similar to each other. And that's called homology modeling. But the first homology model was actually made by hand in 1969. So this is even before the PDB was established. But then, because of the PDB and because of all the structures that became available, many software packages were developed, and I'm sure you have heard of Modeler, you have heard of Swiss Model, and Rosetta and Fire2. And these were all programs that took structures from the PDB and used those as templates to model sequences. And then in 1990s, when more sequence data became available, this is the kind of first paper from actually EMBL with Chris Sander and Alfonso Valencia and others involved in this. And what they realized was, if you align sequences, you can find positions that change and they have a correlation. And so the idea was that if one of these sequences changes, the other one also changes. This was the observation using multiple sequence alignments. And obviously the impact of that, or the implication there, might be that the reason why this has to happen is because when the sequence folds in the 3D, these two things might be close to each other. And so obviously if one changes, that has direct impact on the other, and the other has to change to compensate the change in the first residue. And so this is the coevolution principle, which is then used by all the programs, including AlphaFold, to actually model structures. And this was in 1990s, mid-1990s, so 1994. And then we are here today where we have AlphaFold, which is predicting single chains and structures. But I want to emphasize the two reasons why AlphaFold became possible. So the first reason, obviously, is open data. And I have already talked about PDB as open data, and that is the training set for AlphaFold, but also the sequencing. And through the 90s and 2000s, as we got more and more sequences and more proteomes got sequenced, we now have about 200 million sequences in UniProt. And this is important because now we can understand how the sequences have evolved, because we now have sequences from archaea to eukaryotes and everything in between. And that's important for understanding this coevolution. And so the more diverse sequences you have, the better understanding you can get about evolution and the coevolution of residues. So that's the first reason why AlphaFold became possible. The second reason is CASP, or not necessarily just CASP, but the kind of community-driven evaluation of the progress. So we can think about it as a place where it's everybody who is working on this problem of structure prediction comes together every 2 years. They exchange ideas, they present what progress they have made. So this is a pot where everything is put in, and when you go away from these meetings, you have understood a little bit more about your method, but also what others are doing. And that's a way to progress very, very quickly, because then you are gathering all this knowledge in one place, and that makes it possible to learn really quickly. And so CASP is another reason why AlphaFold became possible, because otherwise everybody develops methods, everybody benchmarks them, and everybody is at the top of that benchmark when they do it themselves. CASP is the place where everybody comes together and the evaluation is really fair and independent. And that's when we really know what's happening. And there are many similar community assessments in life science, like Capri and Cameo and B3R and many others now, CAFA. for functional annotations, etc. I think as we look at this AI and having more and more tools that use machine learning and deep learning, I think we shouldn't really forget about the importance of having these community assessments. And this is for both the scientific community, but also for the funders, that it's important to fund the research in developing methods, but it's equally important to fund these kind of community assessment programs where all these communities can come together. So these are the two reasons why AlphaFold became possible. Open data, obviously, but the assessment is equally important. And then in 2020, the method came out. So this is developed by DeepMind, and I have talked about this previously, so I'm not going to repeat everything that I said at that time. But the most important thing here was the appendix for that Nature paper, which was about 62 pages, which gave every detail of that method. And this is why now we have OpenFold and RosettaFold and everything else who are using very, very similar methods and making progress. But the most important reason why I think these methods are really useful is because they provide these confidence estimates. So this is a metric that the network produces to say how confident the network is in that prediction. And so we can think about it as validation, but this is not external. This is internal network saying what is the confidence in this structure. And there are two of them. And one of them is really popular, everybody knows about it, called the PDBe. And what this is doing is telling you if a residue is happy in its environment. So this is a measure of residue packing, and it's a measure of individual residue being happy in its environment. Okay. The second one is called predicted aligned error. And what this is doing, so if you think about proteins, many times you have all these different domains. And so you want to know if each of the residue is predicted with high confidence. But then you also want to know if there are multiple domains, the placement of those two domains is predicted with high confidence or not. And this is really important, and I'll show an example after this, how it is important to use both these metric. And so the pairwise, the predicted aligned error is telling you how confident the network is. If you take any two residues and their placement in 3D, how confident the network is that those two placements are accurate. And so if the green means it is really confident, and the error in the placement of those two residues is very, very low. And if it is white, it means the error is very high. And so it's just a coincidence that those two residues are present in those two positions in 3D. Okay, so this is just emphasizing the two measures and how should we use them. So the first one obviously is to understand the whole structure, which parts of it are predicted with high confidence, etc., but also the disordered regions. And one of the reasons why the PDBe is so useful is because the analysis have shown that if you have PDBe less than 50, usually it is an intrinsically disordered region. And so in terms of research, this field of intrinsic disordered and how we understand the function of it is going to benefit quite a lot from all these predictions by looking at these regions where the PDBe is less than 50. But even within the regions where you have high confidence, you can start looking at the domains that are present. And this is where I'll show later some of the efforts in understanding the the individual domains, but function related to that, evolution of these domains, and also trying to assign domains to this vast amount of data. So this is one way you can use the PLDDT. But then, as I said, you want to also understand if all these different domains are placed relative to each other with high confidence or not. Otherwise, you are going to get these balls like this, or you are going to have structures with multiple domains, and then you don't know whether this is just a coincidence that they are interacting with each other in this case, or they are not interacting and they are far apart. So what's the confidence in that? So this is where the PAE, as I said, comes in. And so this is just showing an example from the previous slide. So both these individually have very high PLDDT. But if you take, for example, two residues within a single domain, you can see it falls in the green region, which means the placement of it is predicted with high confidence. But if you take residues from two different domains, it's falling in the white region, which means it's just one of the states of that particular protein. It could be anywhere in space, and this is just one example that's available in the AlphaFold database. Okay, so why it is important? Because finally, structural biology is about biology, not structures. So we want to understand biology from these structures. And so if you take a protein like this, you can see that it's a donut shape, there is a pore, and this protein is involved in export from nucleus. So obviously, if you don't take into account any of these metric, you can start speculating that there is a helix that is in that pore, so it's blocking that pore, and it might have function. And the function might be that this is inactive state, something happens, the helix is removed, and then the protein is active, etc., etc., etc. But before we go on that journey, we need to look at the two metric, PLDDT and the PAE. And as you can see, the PLDDT here of that particular helix is low, which means the prediction of that helix and the residues individually is not done with high confidence. But then if you look at the PAE in terms of its placement, you can see that with respect to the rest of the protein here, its placement is not predicted with high confidence at all. So it's just a coincidence that that helix is in that place. And it has no biological meaning unless you actually do experiments and prove something different. So just based on this model, you can't really speculate about the function of that particular helix and what it is doing in that particular position. So it's really important that both these, the PLDDT and the PAE, are used together when you are looking at these models. Okay, so let's see how the database came about. So we had a meeting with DeepMind, the Imbalin team and the DeepMind team at end of March, and we had 3 months to basically produce a database. Usually these kind of things take a year or more, but we worked with the DeepMind team to get everything together, and the database was launched in July 2021. The coincidence being 2021, PDBe also celebrated its 50th anniversary. And so 50 years of PDBe data resulted in this great advance in terms of AlphaFold. We then have had 3 updates, major updates. In December, we added structures for the whole of Swissprot. The reason behind that was these are the proteins that are well studied, have maximum amount of literature available on it, and we understand a lot about function of these proteins as well. And so obviously having structures is going to only accelerate that research. But then I think there are proteins which are equally important but don't have enough structure coverage in the PDBe. And one of the examples being the neglected diseases. And so in January, we added about, I think, 20 proteomes, and every protein in those proteomes was predicted and structures were made available through this database. And then later in the year, we updated the database to basically have structure for almost every protein in UniProt. And so we are now at a stage where we have structure for every protein that we know of and have annotated in terms of UniProt database. And I think this statement is really true. This is one of the most important data sets, I think, after human genome. And this is going to have impact for many decades to come. Okay, so what are we making available in the database? Obviously the predictions, but predictions for all the sequences that fall between 16 and 2700 residues. There is only one prediction, and I'll come back to that later. The accession has a meaning. So this is AlphaFold. This is the UniProt accession that it is for. And then there is this F, which stands for fragment. And what happens is if the sequence is longer than 2700 residues, we chop that sequence in fragments, 1 to 1600, 201 to 1800, etc. And these are the fragments for which we then make individual models available. And so this F stands for that fragment. So you can imagine for Titan how many fragments we have and how many structures we have. But it's all there, and you can access all of it. And people have used it to actually superpose some of these fragments to create a complete structure in some cases, and that's meaningful. Where can you access it? So on the FTP area, we have 49 proteomes available to download. You can obviously use the APIs that we have in the AlphaFold database to get access to all the data, and people have used it to download the whole database, and it's fine. The infrastructure is there to be used, so if you want to use it, that's not a problem. But there are limitations that you need to take into account, again, when you are using these structures. So the first one is most proteins work in complex with something else. It may be small molecule, it may be other protein, it may be nucleic acid. But the database and the AlphaFold method is only for single chain, although people have used it to predict complexes as well, and we'll come to that. But in the database, you have only single chains available. And so you need to take that into account. Having said that, these are two examples where I have compared the prediction with what is there in the PDB. In the first case, the actual protein is a dimer. And as you can see, the prediction actually is a perfect match to the monomer in that dimeric state. The second one is even more scary in a way. This is a DNA-bound protein. So in the PDB, we have structure of DNA and the protein. And I don't think you can tell me the difference between the prediction and the DNA-bound structure. It's actually even there is a loop that goes and hugs the DNA, and even that is in the right place. And I don't know how it happens, but it does create these fantastic models. The second, I think, major limitation is there are no ligands, and in many cases you have cofactors, you have metals that are either important for the structure itself, or they are important for the function, and all these are missing. Having said that, in many cases, you find that there is a hole in these predictions where you can put these ligands. And so even the side chains are predicted in the right place, and all you have to do is to just add that ligand. So that's important. But it doesn't have any of the post-translational modifications either. And that's more difficult to place on this predicted models. So these are all the limitations when you use these predictions. proteins are dynamic, and we have only one model. One of the reasons why we put only one model in the database is because we didn't want to confuse users in thinking that the 5 models that AlphaFold generates are actually about dynamics of that particular protein. So you may find that you have 5 models if you use CollabFold or anything else, or local instance of AlphaFold, and they are different. It's exactly like starting a process with random seeds, and 5 random seeds might produce obviously slightly different things, but that doesn't mean they have meaning. And so if they are different, it's just 5 different things, it's coincidence. But what you would see is, in most cases, those 5 are really, really similar to each other. And this is why we only put one best model in the AlphaFold database. It's, as I said, expected to produce a low confidence prediction for regions that are intrinsically disordered. And so that's information. That is really important information in terms of biological function of many of these proteins. This is a common question we get all the time. AlphaFold is not trained or validated to predict effect of mutations. And so if you take a mutation, just one residue different, and predict a model, in most cases, almost every case, you are going to get exactly same structure as the one without mutation. There are papers which say, oh, the PDBe changes and all that. I can find you 10 examples where that is true. That doesn't make it true for everything. So it's not validated or trained to predict the effect of mutations. And so overall, I think this is a big advance. The predictions are really, really valuable, but it may or may not actually lead to any hypothesis and any understanding of the function of that protein. So when you are using it, be careful, like structures from PDBe as well. All this, what I have said, applies in some cases in PDBe as well. Okay, so what has been the impact? Every continent has used it, and including there was access from Antarctica. I don't know for what reason, but this is one database that has been used from every continent. We have served many people. There are lots of API requests. It's got integrated in many different databases, many papers. It's very difficult to keep track of. So many papers get uploaded on bioRxiv. And there are many examples already where people have used these predictions to do something really important in terms of understanding, for example, the effect of antibiotic resistance or tackling neglected diseases, etc. So let's look at impacts. So I'm not going to go through those examples, but I'm going to go through the general impact and how this is going to change things in terms of research. So the first thing, obviously for a human proteome, PDBe had about 17% of the human proteome residues. We now have almost everything in these predictions, with all the caveats that I have mentioned before. One of the things that it shows is about 30 to 40% of the human proteome is disordered. And this is in line with the expectations that were predicted before, that about 30%, 35% of human proteome is disordered regions. So that's a good place to start looking at if you are interested in IDPs. Okay, but in my mind, I think the biggest impact is change in mindset. We were used to looking at structures that were compact, that nice looking, etc. And that was the effect of the experimental methods that were used to solve those structures. So obviously you can't solve a structure of disordered regions using X-ray or using EM even. And so this is why PDBe had all these nice structures. it's not because proteins look like that or work like that. And so the biggest impact is, I think, proteins are no longer seen as having this well-defined structure. And we have all this spaghetti, and it's equally important for the function of that protein. And this wasn't just a coincidence that we put in this disordered region. This was a conscious decision after multiple meetings to decide what goes into this database. And we decided that we are going to put this whole thing for two reasons. One, obviously we wanted to separate this from what's available in the PDB and highlight that these are predictions. And what better way to do that than having this whole spaghetti in there. The second, as I said, the analysis had shown that that spaghetti is actually a prediction of disorder. And it's equally good prediction of disorder. And so in a way, that also is information. And so this is the other reason why we added the whole thing. And this has already had impact on structural biology. For example, people are looking at these spaghettis, and that tells you which parts of the protein are really nice and structured, and you can then make the sample accordingly. So that's just a start. Obviously, you can take these models and use them for molecular replacement in X-ray, or you fit them in electron microscopy maps, or even look at the NMR data and look at these models. And there are papers which show that these models really fit nicely with the NMR data, sometimes even better than what's available in the PDB. And so there are many ways people have used these models. Obviously, there are many ways people have analyzed it and shown the limitations of these models as well. And so you should look at some of these papers. But I think overall, the direction is that structural biology is changing. What we are going to see is accelerated research in getting structures, understanding the biological function of these molecules. And I have here 3 examples which I thought were really interesting. So this example here is about high resolution structures of complexes from cell extracts. So how do you find what actual complexes exist in the cell? So one way of doing that is to actually get cell extract, isolate some of these complexes, put them on EM grid, and get structures for them. So that's what this paper has done. There are many others now doing something similar for many different proteomes. Obviously, you can take data from multiple experiments, combine it to get structures, and this is nuclear pore. And when AlphaFold actually came about, the database became available, the old model was thrown away. They started from scratch, and within 4 months, they could double the number of components they could fit in this map. Now, this is the kind of acceleration we are going to see in how we understand the biology using structures. The ultimate part of this is where you can actually study structures in situ, in the cell. And that can be done using multiple methods. Obviously, EM, tomography, is one way of doing that. And here, this paper is trying to understand the different conformational states of ribosome when antibiotic binds to ribosomes, etc. So we are going to start seeing structures in situ and structural inventories where we actually understand the whole process in the cell. And that's really exciting. It feels like the same excitement as in 1970s when structures became available through PDB. We are at the same stage. We are at a stage where we are going to restart this whole structural biology revolution, and we are going to understand biology at molecular level. So it's not just about structural biology. Obviously, as we understand more at molecular level, it's going to have impact on biology as well. But there are many bioinformatics questions and challenges that we need to address as well. So one thing, until now, we were used to dealing with about 200,000 structures. Now we have billion structures available. So 200 million in AlphaFold database, about 750 million in ESM Atlas for all the metagenomes. And so we are looking at having billion structures and billion models available to us. So how do we analyze them? How do we have methods that can cope with this scale of data? And you can start imagining if we start having models for isoform and variants and many other possibilities, we are just going to have exponential growth in the number of structural models we have available. And so there are methods like fold-seq which now try and address this for structures, comparison of structures. But there are many, many methods, and we'll see some of those in the next slides, on extending these predictions to protein-protein complexes or even protein-ligand complexes and nucleic acid structures as well. As I said, there are already many, many limitations on the models that we have. So obviously, one research area is how do we address those limitations and how do we get models that are more complete. And so there are efforts, initial efforts like AlphaFill, which add the ligands in the AlphaFold models. And there are also some approaches used to understand dynamics, and we will go through some of these. And so fold-seq is now available, and you can start comparing structures. And one way to actually learn about function of a molecule is not to look at one structure, but to look at multiple structures. They may be structures of ligand-bound, unbound, or protein in complex with other molecules. And then you can understand the mechanistic understanding of the function of that particular molecule. And so similarly here, there is this huge data set now available. We can understand how these proteins have evolved, how the structures have evolved, how the function has evolved. And so one way to do that is to actually cluster these multiple models that we have and try and understand each of these clusters and what's common in there, what function do they do, etc. And so there are two papers, one from Martin, where we were part of that, and another from Torsten Schweder's group as well. There are two different approaches to do this. And there are some really nice outcomes of this. So you can find some of the innate immune system proteins that are common between bacteria and human, or you can find histones that also have a complementary thing in bacteria. And so you can really start understanding how these proteins have evolved and in different species, their function. And this is what is amazing, I think, insights that can lead to experiments to validate some of these things. And this is where we are going to learn a huge amount of biology. As I said, one of the challenges that still remains is complexes. And in the recent CASP 15, almost everyone has used AlphaFold or AlphaFold multimer to predict these complexes, some really challenging ones, very big complexes. And then they have tried to understand how do you score them? How do you identify which is the right one? And this is about understanding the basics of what makes a complex biologically relevant versus a complex that might look right, that might have all the biophysical properties that might lead you to think that this is correct complex, but it's actually not. And so this is not just about scoring, this is also about understanding those basic principles of what makes a complex in a biological environment. And so one of the other things that came out of this whole experiment was that AlphaFold multimer was retrained, and you may have seen that in the news, and it became much better at predicting complexes. Why is that? It's because the AlphaFold multimer was previously trained on all the structures in the PDB until 2016. And many structures from the EM revolution, where we are getting high resolution structures from electron microscopy, were deposited after 2016. And so we now have a lot more diversity available in the PDB for complexes as well. And so when AlphaFold multimer was retrained, obviously it has learned a lot more because of the diversity of complexes in the PDB, and that's what led to better predictions using AlphaFold multimer. Okay, how do you understand dynamics? And this is a paper that's not many people talk about, but I think is really nice concept. And what it is talking about is how do we understand the concept of energy landscape and multiple sequence alignments? Is there an equivalence? Can we think about how the multiple sequence alignment and the energy landscape are related? And so the idea here was you are taking a multiple sequence alignment and you start a process for prediction of structures. It is like starting somewhere on this energy landscape, and then you are going to get a structure depending on where you start. And so what if we manipulate these multiple sequence alignments? Do they have a signature in evolution about dynamics of that structure? So this is not going to work for every protein, but where the dynamics is kind of has left its mark in sequences in the evolution, you are going to start looking at multiple sequence alignments where manipulating them can lead to different predictions, and those predictions might tell you a little bit about dynamics. And so this paper follows up on that, and they have been able to predict the dynamics in terms of fold switch. Obviously, fold switch is an extreme case of change in protein structure, but I think it's a very interesting concept and very interesting experiment to do. And I don't know in how many cases this will work, but I think this shows the direction in which people are thinking about now predicting dynamics. Just one caution, there are many ways in which you can predict multiple structures. And in many ways, they may be different to each other. That doesn't necessarily mean they are all meaningful. And that goes even for the structures that we get from EM grid. So you get multiple structures on an EM grid, but how do you know they are actually meaningfully different states? And those states actually are represented in the cell. So there are quite subtle problems that we need to think about and solve if we want to understand dynamics of proteins or even protein complexes. But I think one field that has huge opportunity here, which hasn't been seen as structural biology field or technique, is proteomics. We can start looking at proteomics in different ways. Proteomics has a lot of data on post-translational modifications. So one way of looking at it is to look at these models, put these post-translational modifications on the model, and see if we can understand more about how these interact with each other, how these things work, what functional relevance they have. So this paper has taken that approach, and they have looked at these three different post-translational modifications using the proteomics data. But also chemical cross-linking is another approach which also can work in situ. So you can have cross-linking in cell, and you can then start looking at what kind of states, what kind of complexes are present, etc. So these things can complement the predictions, they can validate the predictions, or they can be used as experimental data to predict the models of certain complexes, or going forward even dynamics. Similarly, I think there are other methods like HDX which could provide you information. So the important thing to understand here, I think, is depending on your question, you don't necessarily need to have a high resolution structure of the whole protein. And so you can combine different experiment types with these predictions to actually answer the question that you are interested in, and doesn't necessarily need to have high resolution structure in that process. So it all depends on the questions you want to answer. Okay, so what are the challenges in terms of using all this data and using the models? So I'm going to highlight three. One is training, the other is validation, and the third is making structured data useful. Okay, so we now have, as I said, billion models. How do you use them? And this is just showing one example where people are using or abusing the use of these predictive models. So this is fitted in some electron density or electric potential map. This is an EM map, and then deposited to PDB. And what that means is actually PDB might start getting models which are not really the right models. And so we are looking at how to deal with that. And this is because structural biology has become a tool, and rightly so. I mean, as I said, we want to understand biology, and on the way we want to get a structure. And so the methods need to become better and better at getting those structures in a way that users can use them to answer biological questions. And so I think this is a challenge that we are looking at. How do we assess quality of a model? So for example, as I said, there are many different experiments that can be used in combination with these predictions. But how do we assess those things? How do we know the quality of each of these? And how do we know how reliable all these models would be? So that's another challenge which leads to the validation part. How do these various structures differ from each other? So you may have a structure in the PDB, you may have a prediction from different methods. And you may have proteomics data, you may have NMR data, etc. And that might lead to having multiple models for the same protein. But how do you know how they differ and whether they differ meaningfully or not? So that's a question that needs to be answered. And all these, you need to think about who is using these models. So 95% of our users have no structural biology background, and they are using these models. And that's why these questions really become important. And so also, what are the limitations, etc. So we are devoting a whole person to develop training modules and training material so that can be used by users and maybe even teachers to actually teach about protein structures. And that leads me to validation. So obviously the best way we can validate is we can solve an experimental structure, but that's not possible for billion models. And so we need to start thinking about how are we going to validate these. And the way to think about it is AlphaFold-DB is like Tremble. And I don't know whether you know about how UniProt is organized. So UniProt has SwissProt, which is all the manually curated data. And then there is Tremble, which is translated EMBL, which is the database for genomic sequences. And so they are translated, and that's the majority of UniProt. Okay? And then there are rules used that are learned from this manual curation to annotate those tremble entries. And so many annotations you see in UniProt are actually predicted using machine learning or other methods. So what we have here in AlphaFold database is tremble. We have models, but we need to now raise them to a level where they become useful and they become kind of Swiss plot equivalent. But we can't do that manually, looking at every model. And so one way is to start comparing them. But in terms of experimental methods, as I said, many different methods could be used to validate this. We don't have to always use X-ray or EM or NMR. But we need to maybe start thinking about validation in a completely different way. So until now, we always think of validation as a validation of the whole model that we have. And maybe that's not the way to think about it. We have many experiments over the years in literature where people have done mutational analysis and many other types of experiments to study a protein. Right? We can take that, we can put it on these models and ask the question whether the model actually explains the results that we get in those different experiments. And what that means is we are validating a part of a model, not the whole model, a part of a model based on different types of experiments. So maybe we need to start thinking about functional validation rather than structural validation of these predicted protein structures. But it makes it complicated because how do then users understand what part of structure is actually validated and which part is not validated, etc. So there are quite a few complications on how we can explore this and how we can evolve this to make it useful for users. So let's look at how to make it useful. And what I'm going to do is to use PDBe as example and then explain how this can be applied to AlphaFold as well. So I like this a lot because coordinates themselves are just shapes and numbers. They don't have any biology. So you can maybe understand this is a model, right? Maybe because it was in a crystal, you expand the symmetry, but it's still a shape. You have no idea what it is or what biology you can learn from it. And so to actually make these models useful for biologists, what we need is to annotate them, saying this is a ligand binding site, this protein is the spike glycoprotein, these are the interfaces, etc. That's what makes it useful in biology, right? So we need to enrich these models and add functional and structural information so that they really become useful for biologists. But as a biologist, there are some very simple questions you need to first answer. So how do I access models, right? So there may be many. Where do I go? Is the model really good or not for my use? What are the limitations of a particular model? What structure data is available? So is there a structure available for a complex of interest? If they differ, do they differ meaningfully? Do I have to use all of them? Do I have to use one of them? Which small molecules interact, and again, are they meaningfully interacting? Are they necessary for the function of the protein? And obviously about the biological information that can be derived from those structures as well. So in 2019, before AlphaFold 2, we started thinking about some of these issues because in AlphaFold or Cas13, with AlphaFold 1, we had seen a huge progress in prediction. So it was obvious that at some point predictions are going to become really useful. So the question then was, how do we make these data available? So the situation would be that there will be multiple data resources, the PDBe, and then some predictions as well. As a user, you have to know every one of them to get access to it. But a better way would be if all these work together to produce one way of accessing all of them. And that's the idea behind these 3D beacons. So the questions were, how do we track what structured data is available? And how do we standardize the mechanism by which the quality assessment is provided? Because it is equally important to make sure that the quality metric is made accessible at the same time as the structured model. And so we worked with all these different data resources, and it's an open consortium. So if you have a huge database of predicted models or experimental structures, etc., you can join this. And all we have done is very simple. We have agreed on a common API. Every database has implemented it. But what that means is if you come to this 3dbeacons.org and you ask, do I have a structure for this protein? You get access to all the models all across these databases, both experimental and predicted, in one place. And so that's something is already working. But the next step is we need to enrich these models. We need to add the biology on top of these. And we can do that at different levels. So you can do some of these things at per atom, some properties. Some are for a given chain. Some are for a complex, some are for domains, etc. So there is a whole hierarchy, and we need to add information at each of these levels to make this data useful. So we started thinking about PDB and limitations in the PDB. And so some of the next slides are about some of the projects we are doing to make the PDB data useful. So the first question was complexes. And one of the examples I always give is hemoglobin. And I say there is no hemoglobin in PDB. And what I mean by that is we don't ever annotate hemoglobin as a complex in the PDB. There are different types of hemoglobins. The only reason when you go to any of the PDB site, RCSB or PDBe or PDBJ, you get all the hemoglobins is because hemoglobin is in the protein name. And not every complex has its name in the protein names or its components. And so one of the challenges we wanted to address was complexes. How do we identify them? And if we uniquely identify a complex, then we can start comparing, contrasting them, and learn a lot more about complexes. And so we wanted to make it easily findable, and so we need a system for that. And so we worked on it for now 2 years, many different interns and summer placements. But we have now identified every unique complex, and we have a method for doing that every week when PDB is released. We have all the PISA information for every one of those. We have enriched the information on covariation for all the interfaces in all these complexes. And that now allows us to aggregate all the data. And we can now start asking questions about, are there different conformational states for these complexes in the PDB? And are they meaningfully giving us information about the function of that complex? We can also start understanding the relationships between all these different complexes, because you may have a ribosome, and you may have a structure of a ribosome with mRNA, and you may have a structure of a ribosome with tRNA as well. Now, first question is, are these different complexes? The second is, what is the relationship between them? And so we had to do quite a lot of work in terms of annotating all the RNA molecules and trying to understand what is a complex and how are we going to define that. But now we can actually make these relationships. So we can say these are all ribosomes. These are ribosomes where you have mRNA bound. These are ribosomes where you may have other factors bound, etc., etc. And you can now start looking at all those. Okay, so if you go to the PDBe page and search for things, you now can get the name of the complex, so you have a standardized name, you have a accession for it, and so that's unique, and that means if you click on it, you get all the entries that have that particular complex. And all this is, so anything I show on the web page comes from an API. So if you are doing large-scale analysis, everything I show is available through APIs as well. Okay, so the next question is, are there different conformational states in the PDB? And so we are just finishing this project, but I'm going to give this example of hexokinase. And there are 4 different PDB entries, and traditionally, you look at each one of them. And then, obviously, you have forgotten what you saw in the first one by the time you look at the 4th one. And as a biologist, it's very difficult to know which one to look at in the first place, right? So it has 6 different ligands. It's a homodimer. And you can get understanding of the function if you look at the paper. What it says is the sugar binding induces a large conformational change, whereas ADP binding does not. That's the biological understanding that you need when you look at all these models. So the first thing is to understand, are there different conformational states? And so we have now gone through the whole PDB. For every protein, we have clustered the proteins, we have identified different conformational states, in the process created benchmark data sets, and we are at a stage where we can actually show this information as 2 different states in the PDB, not 4 entries. And we are now looking at how do we assign biological meaning to each of these clusters. Again, all this is available for the whole PDB and through APIs or on the FTP area, etc. The third thing we are doing is working with Kat, Scop, and the core team as well now to actually assign domains for the whole of AlphaFold and whole of ESN fold as well. And so we are investigating that and creating one place where all these different domain annotations will be available. And so you can start learning about evolution of domains and all these different proteins and combination of domains found in all those proteins. Small molecules are really important. And so we are looking at small molecules to improve the standardization of the data that we have, integrating information from different databases. So is there bioactivity data for some of these molecules, and how do we link to that? And in the process, sometimes in the PDB, you find a huge molecule broken up into smaller components. And then it's very difficult to match these things. And so we are creating and identifying all those instances. And actually, another important thing we have annotated is all the cofactors, substrate, products. So all the meaningful small molecules in the PDB. And so we are continuing to do that to also identify ones which are just coincidence, so just from crystallization condition, etc. And so by doing this covalently linked components, bringing them together, we are able to identify many more matches to other databases that have more information about these particular small molecules or the biological function of the target. And so you can see some of these things where previously we were not able to identify these molecules as chemically meaningful and biologically meaningful molecules in the PDB. As I said, many of this knowledge is actually in the literature. And so we're working on a project where we want to harvest residue level annotations from publications and enrich PDB data. But we can only do so much. And one of the things that is fantastic is because PDB has been available for so long, there are many groups across the world who take that data and actually analyze it, enrich it, add value to it. And so we decided to have a database called PDBe-Knowledge Base, which is again an open consortium. Anybody can join. it's complementary to PDB. And in PDB, you deposit structures. In PDBe-Knowledge Base, you deposit annotations for those structures. And we already have 34 different data resources from 13 different countries doing that. And some of it might be just a subset of molecules in the PDB, or you may have annotation for the whole PDB. It doesn't matter. What matters is we bring some snippets of it together in one place so that as a user, I don't have to know 400 databases. And so this is not about just taking all the data from everywhere. We work with these databases to take a very small amount of information and then link back to the original source so you can get more information from the original source as well. What do we do with it? We create a knowledge graph. So we create a graph database where we link all the PDB to UniProt, and not just to a single UniProt entry, but all the related UniProt entries as well. We have a lot of information on small molecules, including scaffolds and fragments and their interactions, etc., etc. And all this is available freely for anyone to use. So how do you use it? So just two examples here. So the first question is, are there any small molecules with the same chemical scaffold that interact with the same binding site? And you can write a query in this graph database to get answers. So here is the answer. There is small change here, otherwise the scaffold is exactly identical, and the binding site is identical as well. So you can start learning about how these small molecules interact. But not just that, because there is all this other information available, you can see if any of these residues are conserved, any of these residues are active site residues, etc., etc. And then there are other ways you can obviously write queries and use the database as well. So what we are trying to do in knowledge base is, instead of showing every individual structure, we start aggregating all the information for a given molecule across the whole PDB. So here is an example of Aurora kinase, 169 structures, 153 different ligands bound to this molecule in the whole PDB. And just by going to this one page, you can download all these. I'll show what else you can do, but you can get all this information in one place. Okay, so you can then look at conformational states of this particular molecule. And I don't know if you know the database that Roland Dunbrack has about kinases, where he has classified kinases into this DFG in and DFG out. And this is what you get. So there are two states here. There is DFG in state and DFG interstate. And so these clusters have meaning in the context of these kinases, for example. You can then superpose AlphaFold structure to see what AlphaFold has predicted. Has it predicted the DFG in or DFG interstate, etc.? You can get the RMSDs for all that. You can look at the ligands, and now you are looking at all the unique ligands in the whole of PDB that interact with this particular molecule, right? You can start seeing where they interact as well. So this is all mapped onto UniProt sequence, which means you can see patterns where certain residues interact with many different types of ligands. You can download all the information, but you can also see the superposition of the whole PDB in one view. So you start seeing the pockets where actually multiple ligands bind for a given molecule. So what we are trying to do is to create this infrastructure for future where we bring together both the predicted models and the experimental models, enrich them with annotations, create these knowledge graphs, and make those available on the interface so that biologists can use all this data, and also through APIs so that you can analyze it in the labs through pipelines as well. And I think the major thing here is to then understand what are the similarities and differences between all these different models. Are they telling us anything about the biology? And obviously all that has to be underpinned by training, because that's really, really important. So in summary, I think structures and their biological context are essential for answering biological questions. And the billion structures that we have, they're going to change everything, how we do biological research. But we need to have the right infrastructure in place for that as well. Training is really important, and PDBe is an open consortium, 3D Beacons is an open consortium, so if you want to join, let us know. And we are going to see this time and again in other domains in biology as well. As we have more and more experimental data, there will be new methods, deep learning methods developed to fill in the gaps and understand more of this biology. So with that, I just want to acknowledge the team. These are the resources that we manage, all the funders, all the different consortiums that we are part of. Just an advertisement for a computational biology course at Heidelberg. So it's in December. So have a look, and useful resources. And I'll stop there and take any questions if there is time. And sorry if I have gone over a lot.
Thank you, Dr. Samir, for this great talk. So we are open to questions.
Oh, thank you so much for the fantastic talk. It was amazing, awesome. And I thank you, the organizers, for putting this up together, because it's a fantastic opportunity for all of us that has been so, well, we don't have contact with this much of information, so it's really fantastic. So my question, as you already mentioned, is regarding disorder. As you know, we have all this flexibility and involved, and could you tell us a little bit more in terms of specifically when we look at the predicted structure that you have a single one out, so how we can make sense, how storing this stuff, and
So what is
and the last one is,
Yeah.
can we access and manipulate to have ensemble of these stuff out of the
Okay.
different levels of disorder?
So one of the things that's interesting, and there are again different people already analyzing this, and there are papers already on bioRxiv and in publications. So sometimes when you look at these predictions, you have all this spaghetti, and suddenly you see a helix that's in all this spaghetti. And actually many of these are motifs that bind in a context. And so in the analysis that at least I have read, a couple of them have shown that actually AlphaFold is predicting the bound structure for some of these motifs in middle of all these disordered regions. It's not just that. In many cases, again, this is anecdotal, so not analyzed properly. In many cases, the 3D position of those helices are in the right place for interaction with the partner with respect to the rest of the protein. And so one thing I have seen, for example, this was done using electron microscopy and validated in one case. And then you could find all the other cases where a similar arrangement exists. And in many cases, those proteins actually interact with the same partner or partner in the same family of proteins. And so this is what I meant by there is rich information in these predictions in the context of IDRs. And so that's a huge field that's opened up. Many people are now working on that. In terms of ensembles of these, I think dynamics is not a solved problem in terms of predictions. So obviously, what is going to happen is you look at these, you collect a number of proteins or a family of protein, you analyze the predictions, and then decide to do experiments to actually get the ensembles from that experimental part until we have a really good data set for these, or somebody comes up with a fantastic method for predicting dynamics. That is the only way we are going to learn about dynamics, especially for everything, not just IDRs, but even for proteins or complexes. I don't know if that answers your question.
Somehow, can we couple with some decent force field that would incorporate some of this information besides NMRs and other stuff?
Yes, but that's not, so AlphaFold doesn't have any energy terms at all. So there is only one final refinement done, but heavily constrained, to just make the protein more regularized. And you can see that from so I showed you the ligands, pockets, right? Now imagine if there was a force field. None of those will survive because there are like 4 cysteines pointing to each other without anything there. So there isn't any energy terms, any physics in AlphaFold at all. And so there are other people who are trying to combine dynamics and deep learning methods as well. And that may be the way we will get there. But AlphaFold itself has no understanding of physics.
But in other presentations of yours, and you have cited works when they look, for example, things that are not so disordered like fold switching proteins,
Yeah.
in which they were able to get like intermediary structures.
Yes.
How
So
do they get those?
that's done by manipulating the MSAs, not by incorporating physics there. So no energy terms. That's manipulating the input.
Hello. I understand that most of the AlphaFold was use the PDB as
Yeah.
start. And when you solve the structure with crystallography, for example, you have the temperature terms,
Yeah.
which is similar to what I believe, which is similar to what you say that you call as a confidence residue. So how this residue confidence term that we are using here compares with temperature? Does it compare? Have you ever done?
So the paper from Phoenix guys have some of that information. So if you look at the paper I had on a particular slide.
Do you have an idea just to
So
let me know now?
So it depends, because temperature factors are not just about the dynamics part of it. Temperature factors can be high because your data is not good, for example, or you haven't done the refinement properly. And so in what they have found is in many cases, the loop regions are different when you start looking at using predictions in experimental data. And obviously we know the loop regions are mobile, and so you would expect them to be in different states, right? But most of the rest of the protein which is structured is very much maps in most cases to experimental data, and the electric potential or electron density maps. And so I would imagine that in many cases the temperature factors and the PNDDTs correlate very well, except for the regions which are loop regions where you expect them to be different. But in many cases, then the PLDDTs are lower anyway for those reasons. So in that sense, they do correlate there as well.
One of the structures that you used here was deposited by me.
Ah,
One
okay.
of these structures that you used in this example was deposited by me.
It's good to meet people who actually have deposited some of these examples. Yes.
Thank you, Professor, for your really nice talk, and it was very enlightening. You said that so proteins are very complex in the environments they are in.
Yeah.
So different temperatures, sometimes different pHs. In AlphaFold there is the population state, so that has no physical meaning. It's like a last step. But how this information about temperature, different environments that proteins are in, they are not considered at all?
Nothing is. So what we have here is a prediction of the fold. Okay, there is no folding in anywhere in this, and so any of these parameters that actually affect the protein folding, let's say, none of that will affect this, because it's looking at a sequence and saying what would be the final structure of it and just predicting that. So none of these other things actually are incorporated in the learning.
That's important, because the sulfate bonds, the disulfide bonds, and even sometimes the protonation states are close to what we see. We need to repeat. Okay. Thank you.
Thank you, Dr. Xavier. I would like to ask a question about the conformation. Most biologically functional structures are two different conformations, like you mentioned. And if you study the solid transporters, probably the anaerobic and the realize we have the open-facing conformations, the inward-facing conformations. If you use AlphaFold to predict these structures, majorly it gives you only one conformation.
Yes.
It's more or less like the training data set is skewed, doesn't even work. So how do you get the other conformation?
Okay, so I'll give you example of kinases. There is a both multiple states of kinases in the PDB, and you can bias AlphaFold to give you one or the other. But what you have to, before I say how, what you have to realize is AlphaFold is about predicting a protein structure, not about predicting a conformation of proteins molecule, right? And so all it is trying to do is to give you a structure that it thinks is valid. It has nothing to do with conformations. Having said that, if you manipulate the MSA, as I said, you can bias it. So if you take just the protein sequence and you give it a template, which is one of those conformations. So don't give it MSA, give it just one sequence and a template. It would, in some cases, use that template, and so you can bias it. But that's like saying, I know the answer and I want to see you produce the same answer, right? It's not really helpful in any way. But the fundamental thing is, it is about predicting a structure, and it has no understanding of the conformational states, or even in the training. So I actually removed some of the slides. So one thing people say, for example, is because it is doing this PLDDT, it has a tendency to produce a bound form which is usually more compact in terms of protein structure, which is again not true. It is random what it produces. It can produce bound or unbound form. Actually, in this case, it produces unbound form, not the bound form. And so again, it is random. And so don't think about conformations when you are using AlphaFold. Think about just the fold that you would get the model you get and what you can understand from that. It's not going to produce multiple confirmations. And you can find examples where it does. If you, let's say if you ask it to produce 100 models, obviously if you know the answer, you can find it in those 100 models. But that doesn't mean the method is developed for producing conformational states. You see what I mean? If you know the answer, you can find it if you do enough predictions.
Congratulations on your work. It's really important and critical for our research here. I'd like to ask a very simple question regarding the neglected tropical diseases update you mentioned at the beginning of your presentation. Can you elucidate a little bit more, give more details on what it meant for the prediction quality after you managed to do the update for neglected tropical diseases?
What I mentioned for the retraining was for the complexes. And I think if you retrain the AlphaFold for individual chains, I don't think it's going to produce a huge difference because it already has enough information in the PDB for single chains. What it didn't have was enough of the interfaces, things like that. And so the complexes have improved, but not, so obviously if you have more training, the individual will improve as well. But it's already at the kind of average PLDDTs of 90. And so another 1 or 2 is neither here nor there. And so it's not going to dramatically increase the prediction quality for individual chains if we retrain that.
One last question.
I was thinking more about the computational challenges, right? And for this audience, can you talk a little more about the key parameters that were used for the training the algorithm or artificial intelligence features that are key in that process?
I can't because I'm not expert in that bit of it. So sorry, I can't. I don't want to say something that's misleading.
We're going a little bit away from the computational details. This is what I do for my talk.
Yeah.
But you had told me, right, you didn't use many defined features really to train the algorithm, I would say, from the database, right? I understand the whole PDB data was fed in, basically, and then the...
So it has learned both good and bad. And so sometimes it does avoid bad things as well, and it does produce better models, even for some of the things that are in the PDB.
And just one more comment. If we see something bad coming out, right? I guess eventually we can see something that doesn't make sense.
Yes.
From our biology.
Yes.
So could we also add that in the knowledge?
Yes. So that's the idea. The idea is that we use this knowledge base concept for enriching AlphaFold database as well, and even now on the AlphaFold database, if you know the structure is wrong in some way because you have done experimental thing, you can send a feedback saying this is wrong, this is the reason it is wrong, and this is the publication where you can find the right thing, and we then pass it on to DeepMind, and they are looking at all these things to improve predictions as well. But in terms of going forward, how we are going to enrich AlphaFold database, we are going to follow the same process as PDB, and we are going to have all the annotations go into knowledge base and then feed into AlphaFold database.
Let's thank again Dr. Samir.
Was the talk recorded?
Yes, the talk was